Hello, people. Today's topic is going to be microbiology of dental caries, which is an important topic. So, next. So, coming to definition, dental caries is a biofilm-mediated diet modeling multifactorial, non-communicable dynamic disease resulting in the net mineral loss of dental hard tissues. So, this is a new definition for dental caries, which has been given in 2017. So, coming to theories of dental caries, first we have to know few theories for historical purposes. The legend of worms theory. So, the legend of worms theory was, as per this theory, yeah, the caries has arisen in tooth because of worms which is present inside the tooth structure. And humoral theory, as per this theory, caries is because of difference in the humors between the body. Okay, between the, for example, difference in the balance between the body. And as per Whitel theory, caries arise within the tooth structure as like a gangrene, like without any external cause. It has been arised by itself. Acidogenic theory, as per acidogenic theory, there is some acid which is present which cause tooth decay. That is by the fermentation of the carbohydrates. And parasitic theory is by parasites. Some microorganism is present which is responsible for the destruction of the tooth. Okay, but here acidogenic theory didn't include any parasite, whereas parasitic theory didn't include any fermentation of the carbohydrates. And chemico-parasitic theory, wherein here there is combination of both the fermentation of the sugars to produce acid as well as the parasite as well as the microorganism to produce the caries. Proteolysis theory. As per this theory, there is first proteolysis, that is proteolysis of the organic components, which led to destruction of the inorganic components. Okay, and chelation theory is like first proteolysis occurs, and after proteolysis, with the product of the proteolysis, now all the inorganic components have been chelated from the tooth structure. And sucrose chelation theory, as per sucrose chelation theory, sucrose is present, like whatever we eat, sucrose is present in the tooth structure. This is going to chelate all the inorganic substances. Okay, Levine's theory, as per Levine's theory, we are going to have a plaque. We are going to have a dental plaque, and with the help of a critical pH below 5.5, there is going to be a imbalance between the plaque and the anti-tooth structure. Like there is going to be in and out movement of the ions, calcium and phosphate, between the plaque and the tooth structure, which could lead to caries. So, yeah. But what is the actual cause? What is the actual cause for this caries? Is going to be four things. One is the host, that is us. Another one is a cariogenic biofilm, and fermentable carbohydrates, and time. Okay, time has been added as a fourth component after other three components. So, yeah, and this is all as per the acido, like both the fermentable carbohydrates as well as the cariogenic biofilm. Okay, as per acidogenic theory. And also we have a primary modifying factors and secondary modifying factors here. So what is primary modifying factor, which is nothing but the salivary flow rate, etc. And secondary modifying factor is going to be the behavior attitude of the host. So yeah, now as per our topic, we're going to take only the microbes. The microbes and their characteristics, Streptococcus mutans and the local immunity, and the adherence of the Streptococcus mutants and the plaque formation. Why Streptococcus mutants alone? Because Streptococcus mutants is the primary reason for the production of for producing caries. Okay, what is the microbes and characteristics? Is we know that the microorganism within few weeks after birth, it's going to be get inoculated into your mouth. And Streptococcus mutants, because of it, local immunity is produced within 1 month after birth. Okay, local immunity is nothing but salivary IgA. And after that, we have to know how the Streptococcus mutants adheres to the tooth structure and how it forms plaque. So for that, we have to know some of the characteristics of the mutant Streptococcus. First is about sugar transport. So the Streptococcus mutans is capable of sugar transport, so that it could be always well nourished. Okay, all types of sugar transport could be undergone by the Streptococcus mutants, and it can produce acids which could actually lead to caries. Okay, which it could lead to caries. And aciduricity is nothing but it can survive in an acidic environment. That is also an important thing, because caries, for prediction of the caries, the bacterium should have this property. And it can produce extracellular polysaccharides. This extracellular polysaccharide is substance which is going to adhere to bacterium together. Okay, so for the adhesion, this extracellular polysaccharide is important rather than digestion, and that is produced by the Streptococcus mutans. And also IPS production. IPS is nothing but intracellular polysaccharide. This is nothing but it's like a storage house inside the inside your bacterium. Suppose if the bacterium didn't get any carbohydrate outside for survival, it can use its IPS and still it can produce acids. Then antigen 1 and 2. This antigen 1 and 2 is needed for to get itself in a like attached to the plaque, okay, to the biofilm, to the pellicle, so it can get itself attached to the pellicle, thereby it can stay inside the tooth structure. And also other enzymes like glucosyl transferase, fructosyl transferase, fructanase, and lactic dehydrogenase. All these enzymes are important. This lactic dehydrogenase is only going to produce lactic acid, thereby forming leading to tissue destruction. So, okay, all these things we just saw about Streptococcus method, that is only the reason for caries. No, there should be a biofilm for the production of the caries. What is this biofilm? Biofilm can be defined as a sessile multicellular microbial community characterized by cells that are firmly attached to the surface and enmeshed in a cell-produced matrix of extracellular polymeric substances. Nothing but it is going to have a lot of microbial community, not really one thing, a microbial community which is just firmly attached to the tooth structure by a self-produced matrix of the extracellular polymeric substances. Let's see what about the stages in the biofilm formation. First is going to be the pellicle formation. Pellicle formation is nothing but, what is pellicle? Pellicle is nothing but a glycoprotein which is from the saliva. Layer of glycoprotein is going to be attached to the pellicle, and after pellicle, initial adhesion. All the planktonic microorganisms. I'll just tell you what are the microorganisms which are going to be initially adhered. They are going to be adhered to the pellicle by forces, by very light forces. Okay, initial adhesion, and then maturation. So after initial adhesion to the glycoprotein, which is present in the pellicle, now maturation is other microorganism, other advanced microorganism, other secondary invaders is going to be attached to your primary invaders. Okay, thereby this could mature, forming a concave appearance. Okay, and after now this mature plaque is very, very difficult, and this is the plaque which could lead to your caries. Okay, which could lead to your caries. These are the microorganisms which could lead to caries. And after maturing, now it is going to disperse. So dispersion could be like single cell dispersion. Why dispersion? Suppose if there is not enough nutrients which in the plaque, now the role of the plaque, there is nothing important, since there is no nutrients in it. Now it could disperse. Dispersion could be single cell dispersion, or asperse loving, like a mass of tissue could get dispersed by itself. So first, this is the pellicle. We can have planktonic cells. This gets attached to itself with the help of low forces like hydrogen bonds, or very minimal forces, van der Waals forces, etc. And after that, high affinity specific adherence takes place. That is nothing but very strong forces like ionic bonds between the planktonic cells, and the planktonic is nothing but they are benign microorganisms. Okay, benign microorganisms which are present. They get attached to the pellicle with the help of strong forces. And the next step is, upon this planktonic microorganism, other microorganisms get itself attached. How this other microorganism get itself attached is because of the polysaccharides. Every microorganism is going to have polysaccharide. This polysaccharide is going to help in the attachment of other higher forms of microorganisms. And also I have to know about few stuff. I'll just tell you in detail. So this is a mature plug. You can just see here you have an aquatic pellicle and you have a planktonic microorganism, which is what is firstly adhered to the tooth surface. Okay, see the streptococcus. All streptococcus are planktonic. All they are early colonizers. Okay, all capnocytophagia, all Propionibacterium, Veillonella, Echinella, Prevotella, Actinomyces, Neisselandia, everything is going to be your early colonizer. So this gets itself attached to Fusobacterium. Fusobacterium is a co-aggregated species which is going to aggregate all the early colonizers as well as the late colonizers. So what is the late colonizer here? Aggregated bacteria, Actinomycetum, Comitans, Prevotella intermedium, Eubacterium, Treponema denticola, Porphyromanus gingivalis. Okay, all these microorganisms, you can just note it down. They are late colonizers. They get attached to the early colonizer with the help of a main microorganism, with the help of a main co-aggregator species called as Fusobacterium nucleatum. Fine. Now, okay, we just know about the structure of the plug and how it is formed and all that. Now, whether they communicate with themselves. Yes, this biofilm could communicate within themselves with the help of a metabolic communication. What is this metabolic communication? Is every microorganism is surrounded by extra polysaccharides there, and these extra polysaccharides could be a source for communication, like nutrient need for a bacterium, for oxygen need for a bacterium. They are all called as a metabolic communication that is possible with the help of an extra polysaccharide which is present around the tooth structure. For example, I'll give you an example. The Fusobacterium nucleatum and the Prevotella intermedium could survive in acidic environment, whereas this Porphyromanus gingivalis cannot survive in acidic environment. What they both do is they excrete ammonia, and this ammonia is going to make the medium more alkaline, thereby Porphyromanus gingivalis could survive. Okay. This is all called as a metabolic communication, but there is not only that these microorganisms help themselves, but also they compete themselves. This competition could be, I have to remember, three stuff. One is bacteriocins, all microorganisms which in caries development they are able to produce bacteriocins. So these bacteriocins act on the other microorganism for competition purposes. Okay, bacteriocins can have an effect on each other. So whatever each bacterium produces. And the second one is going to be quorum sensing. Quorum sensing is nothing but when, suppose if there is a harsh environment, some signaling molecules are released by each bacterium, each species specific bacterium, so that they can communicate by themselves to act in a specific behavior. See, this quorum sensing, they are signaling molecules which are synthesized, and after synthesis, it could be recognized by other bacterium. Okay, and thereby it could give response, virulence, bioluminescence, other group behaviors, whatever group behaviors are needed to wall of that harsh environment, this bacterium could do together because of this quorum sensing. Okay, quorum sensing, you have to remember stuffs like auto-inducers, 1, 2, etc. For gram positive, it is separate. Okay, gram negative, it is separate, and together also it is separate. Gram positive and gram negative. So what molecules they release? Okay, so auto-inducers, they are going to play an important role. And after quorum sensing, other thing is competent stimulating peptide. This is also a competition of the biofilm, wherein suppose if there is a harsh environment, and in that harsh environment, if the microbes think, in the caries microbes thinks like they cannot survive, they can release some lysozymes within themselves and they can go a cellular suicide. So this is possible with the help of again signaling molecule that is competent stimulating a peptide, and also this could transfer gene, okay, genetic transfer. So all these stuffs occur. So we have to know about the competition which is occurring, about the quorum sensing, extra metabolic communication, and also about aggregation and co-aggregation. Aggregation is nothing but one bacterium gets adhered to other bacterium with the help of a polysaccharide which is present in the walls. What is co-aggregation? Co-aggregation is, example, Fusobacterium, wherein one bacterium is going to have so many receptors around it, so which helps in the attachment of the two bacterium, like early colonizers and the late colonizers. That is only co-aggregation. Okay, so everything, all the bacterium adheres together only with the help of polysaccharides which is present around their body. So yeah, we know, we just understood what is biofilm, which is going to cause caries. Since it is a microbial aspect of a topic, we have to also know what is the specific interactions in this biofilm which cause caries. Okay, this is only for caries stuff. Okay, see, we have to know that the Streptococcus mutant could inhibit a lot of other bacterium. They're capable of inhibiting a lot of other bacterium. Also, they are capable of facilitating few bacterium. Okay, the Streptococcus mutants could inhibit a lot of other Streptococcus bacterium, and wherein it could facilitate the growth of lactobacilli. Okay, and the Streptococcus sanguinis and the Streptococcus mutants, they both inhibit. I'll just tell you this thing alone in detail later. Okay, and this whereas Porphyromonas gingivalis is facilitated by a lot of other bacterium. Thereby, I just told you already, right? Like they cannot survive in acidic condition, and that's why other bacterium is going to help them. And this Fusobacterium nucleatum is also facilitated by actinomyces. Okay, this is going to facilitate Porphyromonas gingivalis and Streptococcus oralis. In all these things, we have to know that one interesting factor is that between the Streptococcus mutans and the Streptococcus sanguinis, whereas Streptococcus mutants could inhibit the sanguinis directly by the production of the bacteriocins. Okay, by the as we all already I told you what is bacteriocins and stuff. Okay, they can produce a lot of bacteriocins and they can inhibit that, and also they can produce acid and inhibit, whereas Streptococcus sanguinis could inhibit these mutants by the production of hydrogen peroxide and faster cell division. Okay, now we have to know what is we know how the bacterium is formed and that this thing, but there are few bacterium which are specific for specific sites to form caries. For example, if we are going to, if there is an enamel caries and if it is crown surface caries, the organism which is responsible is mutant Streptococcus. Okay, this one you have to know. Okay, this definitely, which is know about the biofilm and the communication, and also about which bacterium is going to cause what. So we all know that mutant Streptococcus is going to initiate the caries, and lactobacilli is going to facilitate the caries in the crown surface, not only in crown surface. Mutant Streptococcus is going to initiate caries in almost all surface of the tooth. And whereas in nursing bottle caries, we have other organisms also like actinomyces, lactobacilli, and Veillonella species. Because for the lactate formation, what is nursing bottle caries? Suppose if the babies are given bottle for a long amount of time, okay, long term. Then coming to root surface caries, again we cannot tell that only if the mutant Streptococcus is present it will be caries. It could be also, even in the absence, caries could occur, but this is the main organism. Why? Because we saw its properties. There are a lot of properties which makes it to survive, thereby helping in the attachment of other organisms. But it could only initiate, progression is made by other bacterium only. For example, in root surface, you have to know actinomyces is a very specific organism in the root surface caries. Streptococcus sanguinis and lactobacilli, as we saw already in crown surface, but we have to add actinomyces in the root surface caries. And subgingival caries is nothing. There is nothing different in subgingival. If you're going to have subgingival caries, what bacteria will be present there is extension of the plaque community. There is Bacteroides melangenicus, which is a pigmented bacteria that will be present more in the subgingival caries. Now coming to dentinal caries. So now enamel caries, just we can stop with two most common bacteria, mutant Streptococci and Lactobacillus sanguinis, and also actinomyces in case of root surface. Whereas dentinal caries, we are going to have more and more bacteria. It is facultatively and obligately anaerobic gram-positive bacteria belonging to genera Actinomyces, Bifidobacterium, Eubacterium, Lactobacillus, Parvimonas, Propionibacterium, and Rhodia. You can just remember these are some of the not early colonizers, late colonizers, which is going to be established in the dentinal caries, all facultatively and obligately anaerobic gram-positive bacteria. Now, after dentinal caries, the next step is going to be bacterial invasion into pulp. So pulp, it is going to have a lot of pigmented bacterium, black pigmented bacteria, Prevotella intermedia, Prevotella. I have to remember the Prevotella species, which will be definitely a stuff in your late inviters. And then, and the one interesting stuff is like there are few bacterium which are associated with certain things in the pulp. Okay, suppose if the bacterium has invaded into the pulp, in that time, if it check in dentin, you're going to have Actinomyces odontolithecus and Bifidobacterium species. Suppose if there is an abscessed pulp, that is after caries only, okay, the caries invasion in the pulp. If there is an abscess, if we isolate, we can say more amount of Porphyromonas endodontalis and Porphyromonas dentalis. You can just remember by the term dentalis and odontalis and dentalis. And in necrosed pulp, you can see Propionibacterium, Eubacterium, and Fusobacterium species, which is also almost rhyming stuff, and linked with pain. So linked with pain, if the patient's complaint is pain, okay, you can just see Peptostreptococcus micros, and also Porphyromonas and Fusobacterium. Okay, Fusobacterium is there here. Fusobacterium is there in necrosed pulp also. In abscess to pulp, it is going to be endodontalis and dentalis. Okay, now about the application aspect. So what is it going to like why we have to know about the microbiology? First of all, see, we can just identify the caries risk of the individual by knowing the microbiology of the caries. Like there are there are few tests which have been invented to find out whether the patient is having a high risk for caries. Thereby appropriate preventive measures could be taken even before the actual production of the caries. What are those tests? They are lactobacillus count test, dentocult lactobacillus, Sneeder's test, Albin's test, swab test, mutants group of streptococcus screening test. So these tests could be done to just find out, like not for all individuals. Suppose if the patient can't clean their tooth, they have some they are handicapped, or they have a lot of other caries, or if they have historical stuff. If your mother is having more caries, you can just check, or if they're fed for long amount of time using nursing bottle, we can check. So in that condition, during pregnancy also, just to like not for every individual, for few specific individuals, this could be used to check whether they have a high risk of caries. Okay, and then one more thing we have to just no, this is also an application purpose. Why we have to know caries. Dental caries from a molecular microbiological perspective. Molecular microbiology is nothing but we are going to isolate the exact organism which is present in the exact disease. Okay. So there are many microbiological methods. For example, in the caries also, we can just isolate the exact organism which is going to cause caries. Why we have to isolate? Thereby we can give treatment specifically. Okay. So there are like the caries could be treated, prevented even before the actual cause. That is the reason why we have to know this. Okay, there is something called as a medical management of caries. Suppose if we get a topic we can discuss, wherein we can manage it without cavity formation, without doing cavity preparation, and without doing a filling. Okay, for that purpose we have to know the organism. So here this is the same way from molecular purpose, descriptive and approach and the functional approach. What is descriptive approach is? We are going to see the DNA. We are going to find the exact organism using the DNA. One is 16S rDNA approaches and metagenomics. And functional approach, in the functional approach, we can identify which organism using the expression, using the RNA expression, that is called as metatranscriptomics. Using their protein content, we can identify the exact organism, that is called as metaproteomics. Using the metabolites, what is released after metabolism, that is called as metabiomics. Okay, just the few you can remember names, don't want to go into much detail. And other application is caries vaccine. Okay, so what is this caries vaccine? Caries vaccine is also one of the medical management wherein for other purposes, like we take vaccine for caries, also we can take vaccine to prevent caries. That is, if the patient is undergoing a radiation treatment, we can see how badly the teeth will be affected after that. So these caries, they could be prevented by this, but caries vaccine is still under research, however. Okay, so this caries vaccine, how they act is by interfering with the adherence, colonization, and dissemination, altering the ability to produce acid, reducing its stickiness by altering the polysaccharide mechanism. This vaccine does some effect to the microorganism, thereby preventing its accumulation, colonization, ability to produce acid. It's going to change some all these things. Okay, so what there are two immunization. What is called as active immunization, wherein we are going to give some what antigen, and thereby antibodies are released from our own body. That is secretory IgA. IgA is the main antibody. Okay, so it's active. Passive immunization, we are going to what monoclonal antibodies. We are going to inject antibiotic itself to the individual. And under active immunization, we have so many routes. One is oral, intranasal, tonsillar, minor salivary gland, rectal. Okay, so all these first five routes are found to be much more, what to say, much more better than other active gingival salivary systemic routes and passive dental immunization. The first five routes is going to be much more better. Now, this caries vaccine was initially produced for lactobacillus, but now they are just after that for streptococcus, and now even streptococcus sobrinus, we have they are just doing research to produce this vaccine, so that just this vaccine is going to help you by secreting more amount of IgA if it is going to be active. Passive, you're going to give IgA itself directly. Okay, so that's it. We have few questions regarding whatever the class has taken. So first question is, which bacterium is associated with endodontic pain? Fusobacterium, Streptococcus, Lactobacillus, Actinomyces. Any guess? Just can just guess. It's very easy. Whatever we taught today. See, endodontic pain. You have to remember it's Fusobacterium. Okay, certain bacterial combinations are associated with endodontic clinical symptoms. We have to remember Streptococcus micros, Porphyromonas, and this Fusobacterium is going to be linked with pain. Your second question is, which can induce the cellular suicide in the microorganism under stressful condition? Auto-induces 1, auto-induces 2, auto-induces 1, competent stimulating peptide, and stamps. The answer is going to be competent stimulating peptide. As I already told, this is one of the competition in the producing signal producing molecule in the biofilm, which is where your cells could undergo suicide under stressful condition. Next question, which among these is one of the best known co-aggregation bridge species in the Caries biofilm? Streptococcus, Fusobacterium, Lactobacillus, and Actinomyces. This is a very easy question, so I want to answer for this. Which is a co-aggregation bridge species? Just a guess from all the four options. Co-aggregation bridge species, you have to remember it's Fusobacterium. Okay, Fusobacterium, just don't forget this name. It's very important in the biofilm formation. So Fusobacterium is the organism which is going to cause this co-aggregation stuff between your primary and secondary invaders. Next is identify the correct order in the biofilm formation. Pellicle formation, initial adhesion, maturation, dispersion. Initial adhesion, pellicle formation, maturation, dispersion. Dispersion, pellicle formation, initial adhesion and maturation, or pellicle formation, initial adhesion and dispersion. So the answer for this is, first there will be pellicle formation, initial adhesion, maturation, dispersion. We know this, right? So answer A is the correct stuff. First there should be pellicle for adhesion, then adhesion, thereby forming maturation of the biofilm, and then it could disperse on itself. The next question is, tolerance of low pH in a bacterium is called as what? Aciduricity, acidogenicity, extracellular polysaccharides, intracellular polysaccharides. The answer for this is aciduricity. If the bacterium could survive in an acidic condition, it is called as aciduricity, wherein if the bacterium is able to produce acid, that is called as acidogenicity. Okay, if they have special specific biochemical attributes enabling them to survive and grow at a low pH values. So that's it. Thank you so much. If you have any doubts, you can ask. This is little dry, but I think I had made it little interesting. I tried to make it interesting, but it's a very small topic. If you have any doubts, you can ask, or we can find it. Thank you.